So next is Heather Echevers, who's going to talk to us about initiatives in France.
Hello, thank you very much to the organizers for inviting me to represent our French initiative, which is sponsored by the INSERM, which is the French National Institutes of Health, to coordinate our efforts into understanding human developmental cell differences, and to hopefully articulate with the HCA and to participate in your wonderful collaborative efforts. So the INSERM decided to sponsor this effort because human congenital diseases and malformations affect a really large spectrum of the population, although we don't always realize it because not everybody's born. But beyond that, a number of chronic and late onset adult diseases also begin during prenatal life. So we found that there was really a need to support the individual efforts that have been going on in France for many decades, studying human embryonic and fetal development and putting that into relationship with other developmental biology expertise that are developed in a fairly local manner, and to try to bring us together with the computational biologists and the people who are developing these molecular techniques and the more cutting edge technologies so that we could all work together. So the structure of the project is obviously centered around the analyses in the middle there that have to do with single-cell RNA sequencing, but not only as a technology in order to attribute cell identity, but also in capacity building for bringing together our different resources in how we collect cells, tissues, and organs at a national level, and then not only conduct the single-cell analyses, but also validate them and put them in relationship with studies that are underway relative to cellular mechanics and their dynamics, and to reposition these cells within space and time, so 3D and 4D mapping. And then finally, of course, part of this project is to make available the data not only obviously among the partners, but to the wider scientific community, and to make an effort in a prospective manner to ensure that the data is compliant and compatible with other initiatives, and in particular with the HCA. And so one of our particular strengths in France is that we are able to obtain not only project-oriented organ and tissue primordia, but also many hundreds of intact human embryos and fetuses that can be used not only for our projects, but that we are setting up a framework to make these resources available to the wider international scientific community. So I was very happy to hear about the working group on ethics, because this is a major component of how we're going to be able to distribute these resources more widely. So this is a pilot project. It's funded for the next 3 years. So one of the very specific aspects to our project, which I don't think is really accounted for in the other developmental atlases at the moment, is that we've foreseen to set up a cryobank that's devoted to pre-implantation embryos. And this is because there are a number of IVF clinics in France from which many embryos are donated to research. But however, we noticed that a lot of that donation is not particularly coordinated, and there's a lot of waste, actually. And so 2 centers within Paris have been funded to recover a certain number of these embryos and under very reproducible conditions and make them available to research. And we also have a funding of a pilot project that's underway using these pre-blastocyst up to blastocyst stage embryos to study the mechanical properties of the individual cells within these embryos and relate these properties to their molecular characteristics as a proof of concept for conducting similar studies based on cellular mechanics on later developmental tissues. And you know this work very well, but these wonderful studies that have been taking place within model organisms in zebrafish and in mouse in defining the cell types and the cell states that are important for development, we felt the great need to apply our expertise to starting an atlas or contributing to an atlas that has to do with human development. And so we've picked a few pilot projects in order to both piggyback on efforts that are already underway. For instance, you heard about Pascal Barbery's work on the lung, and so he's conducting a project, which I'll describe briefly. I'll just have to skim through these for the question of time, but I did just want to emphasize the fact that we've already foreseen that it's quite important for us to integrate both genders as far as, sorry, both sexes, I can never remember which one I'm supposed to say in English, and also a number of different organs and different developmental time points. So we've picked the ones that are the most frequent and easily represented, but we're planning to extend beyond this in the future. We're also doing some technology comparison studies that are specific group dependent, using techniques which hopefully will be able to maximize the amount of money that we can devote to these different efforts, depending on the organs and the different specific questions that are relative to those tissues and organs. And then, as I mentioned, the idea of confronting not only single nucleus RNA sequencing, but cross-referencing that with ATAC-seq and comparing fresh and frozen procurement protocols so that we can optimize how our collection centers articulate with our library preparation and our sequencing centers. And so, of course, ultimately, all of these molecular properties, we would like to map them back onto the tissue, so we can do that with our in situ hybridization techniques, both in 2D and in 3D. But we are also very interested in developing these tissue clearance methods and continuing to work with them in order to do some protein mapping as well in 3D and to reconstruct either whole embryos or organs in such a way that we can explore specific aspects of their cellular properties in context. And so finally, the third part of the project has to do with the data accessibility, making our data accessible among ourselves in order to know which samples are available and at what time and in what condition, to link this information with the molecular data that we would generate internally, and then of course we'll deposit within repositories that will make it accessible to the greater community. And to ensure that that happens, because it's not enough to say that we want to do it, we actually have to do it. So that gives you an overview of our HUDECA project. These are the different centers and laboratories that are involved. And I did want to mention that we are planning on hosting a symposium in any case, which appears to articulate very well with other proposals to do the same thing. So we will each take turns and hopefully build something quite productive, all of us together. So thank you very much.
Thank you. Any questions?
That was great.
Thank you.
What's your plan for metadata about dysmorphology and trying to
So at the
various
moment,
scales?
Absolutely. So we are going to recruit what comes in, but of course, first trimester recruitment does involve that much more dysmorphology than you would see at birth, for instance, or in pediatric populations. So our plan at the moment is to keep everything that comes in, so that we're establishing this biobank, but that the second and third parts of the project are going to be devoted to normal tissue. And normal we are going to define based on molecular markers relative to aneuploidy in particular, and to morphological landmarks.
Are you thinking of some, say, 3D, fast 3D imaging or something per sample that where, other approaches to quantitating dysmorphology might be applied?
We're a little more, low throughput at this point. So part of the criteria for staging and for assessing whether embryos are normal just has to do with typical binocular microscopy photography. But in essence, what we need to do is to document why we have said that a given embryo is normal. And so we want to base that both on morphological criteria and on this molecular analysis, which is superficial, but we want all of that metadata to be attached to each sample, which is then analyzed so that it can be accessed in both directions. Thanks.
Okay.
Thank you.
Thank you very much.